In this video, we'll be talking about how to identify different structural features found in peptides. So first and foremost, this is with the assumption that you know the structures of the 20 commonly occurring amino acid side chains. So it's important that you not only know those structures, but you'll also need to know both the 1 and 3 letter abbreviations for each of the amino acids, as you'll see as we go through this. So again, it's not always fun memorizing things, but you must know these structures or at least be able to recognize them. When we talk about peptides, what we're talking about is linking multiple amino acids together using both the alpha carboxylic acid and alpha amino group on the amino acids. With our peptides, they're going to start with the N-terminal end or a free amino group. So there's our free amino group. And it runs, goes from the amino terminal end to the carboxy terminal end where we have that free carboxylate at the opposite end. We'll also look at some common modifications that can occur to both the N and C terminus, and you should be able to pick those out as well. We mentioned that with our peptides, we're linking these through a condensation reaction between the carboxylate, our carboxylate group on one amino acid and the amino on the next. I'm circling here one of those peptide bonds. Peptide bonds are amide bonds. You can see we've used this. This was the CO2 group on our first amino acid in the peptide chain, and that's linking with that NH2 group on the second amino acid in the chain. So in this peptide we have 4 different peptide bonds present. We have 5 amino acids. You always have 1 fewer peptide bond than amino acids. So on this peptide, you should be able to identify the amino terminus and carboxy terminus. It's easy in peptides that aren't modified. You should also be able to highlight the peptide bonds. In this instance, it's a little bit easier since it's already done for you. And then you should be able to either identify specific amino acids, like if I, for example, said identify the tyrosine side chain, you should be able to identify that and say a hotspot question. Or if I had that group circled, you should be able to tell me that that's the side chain of a tyrosine. All right, the other thing we want to be able to do is provide the sequence for the peptide that's shown. So in here, you always start with the N terminus and work your way to the C terminus. So starting at the N terminus, we see our first side chain here, the CH2OH is a serine. I'll put the sequence here at the bottom. So we're going to have ser as our first peptide. Next we have a hydrogen, which is glycine. That's followed by, we've already talked about our tyrosine. Methyl is the alanine. And then kind of this V with an extra CH2 is leucine. So you should be able to provide the sequence of that peptide as well. Normally with a sequence, I have you provide the 3-letter abbreviation. Once you get to larger, either really large peptides or proteins, it's more common to see the 1-letter abbreviations used. So we'll work through a few more examples. So here we first want to circle the peptide bonds in the tripeptide. So we know since it's a tripeptide, we're going to have 2 peptide bonds. And so our first peptide bond is there. That's our second peptide bond. So again, those are amide bonds along the backbone. The next part of this is to identify the amino acid sequence for this peptide. This is where we want to go through and identify each of those amino acids. We're going to have 3 of these in the tripeptide. Some folks find it easiest, to make sure to identify their alpha carbons, to know which side groups, to help identify where the side groups are located. All right, in this peptide, we look at our first, remember we always go from the N to the C terminus. On our first alpha carbon here, we have the side chain of a valine. And I cannot get that line to reproduce, so I can't get IT to help me. The next, if we go to number 2, we have a hydrogen, so that's glycine. And then finally, we have just a methyl group, so that would be ALA. So that's a valine, glycine, alanine peptide. All right, we'll go again with this. Again, here's our N terminus. That's where we're going to start. C terminus is where we're ending. All right, so first amino acid side chain is going to be right here, our tyrosine. Following the peptide bond, our next amino acid is going to be glycine. Followed again, our next amino acid is another glycine. That is going to be followed by phenylalanine. And then our last group is a methionine. So that's how we go through and identify the amino acid sequence for the peptides. We will look at some modifications that can occur at the N terminus and C terminus, which complicate the identification. The main N-terminal modification we've looked at is this pyroglutamate residue, which is just a cyclized form of either glutamate or glutamine. So you can see that shown there. Looks a little bit similar to proline, but you can see it's an amide within the cyclic structure. The other common modification we've seen occurring, and that was at the N terminus, the modification we've commonly seen at the C terminus is the conversion of that C-terminal carboxylate, which is negatively charged, converting that into a neutral amide. So with our oxytocin structure here, you can see that NH2, where they have it in black, that's an amine that has been added onto the carboxylate. So that does, maybe in some ways complicate that identification, but that is the C-terminus of that peptide. So we can take a look at our tripeptide TRH shown here, and we are going to take a look. We can identify our peptide bonds. There's our first and the second peptide bond. What we're going to do with our sequence, we don't want to be fooled. We know a common internal modification is the formation of the P-glue. So that is our first residue in this peptide chain. At the next position, we have this histidine side chain. And then we have our proline. And we note that the proline C-terminal amino acid has also been modified with a NH2 group, so we've converted the carboxylic acid into an amide. So when this occurs, folks usually then just write an NH2, indicating that the proline isn't a free carboxylic acid, but rather it's the amide form. All right, here we're going to take a look at oxytocin again. Just trying to get practice identifying the termini. So here we have the glycine. So we'll first identify our N terminus. There's our free NH2 group coming off of the alpha carbon. Our C terminus is going to be that modified glycine, where it's the amide instead of carboxylic acid. The other thing unique, the other structural feature you might be asked to identify would be the disulfide bond. This is the bond that occurs where you're forming a sulfur-sulfur covalent bond between two cysteine side chains. That's also present on oxytocin. that results in the cyclization of that right half of the peptide. Now, with something like this, I could also provide the structure where the abbreviations, the identification of the amino acid is not already done, and ask you to identify, a tyrosine side chain, ask you to identify a leucine. This is something you can use as good practice where it gives you the key. You could block those out and see if you get the correct amino acid. All right, here we go again. As I said, here's one where we've blocked out the amino acid labels. I might ask you to identify the phenylalanine side chains. There's going to be 2 of them there and there. On this peptide, we could also, have you identify the N and C termini. Our N termini, a little bit easier in this one since it's not modified. That's shown there. C terminus, again, we see that common NH2 modification. You should be able to take this, go through and identify all of your side chains. We won't do the whole peptide, but we started. So we have an arginine side chain, proline, your lysine, we got another proline, and so forth. So you could walk through and identify the different amino acids on that peptide. Here's some additional C-terminal modifications we've looked at in blue. So I've N-terminal are highlighted in red. All of those are the P glue. And then with the blue, those are other C-terminal modifications. They're still amines, but it might not simply be an NH2. It might be some other type of amine where you're converting the carboxylic acid into an amide. You can see we've done that with all of those structures. And the last example we'll take a look at is having you identify, so given a peptide analog, having you see if you can relate the structure of some drug or drug-like molecules into the amino acids. You might be asked to identify the side chain that looks most like lysine. That would be option B, looks the most like the side chain of a lysine. We could ask what side chain most resembles tyrosine, right? That would be our option E. We then have our A would be our arginine. And we've already looked at phenylalanine before, but D is our phenylalanine structure. Structure C is a modified C terminus. It could also be used to mimic the side chain of, say, a glutamine or asparagine, where you have just the amide NH2 present. That will wrap up looking at identifying the structural features of peptides.